Hello student, now we are going to discuss male reproductive physiology. So in male reproductive physiology, what are the different parts of the reproductive system that we are going to discuss? So all of you know that the main part of the male reproductive system is the testis, which is connected with this epididymis, as you can find out here, epididymis. Then this epididymis is converting into or connected with the vas deferens. So this is the long vas deferens. This vas deferens will ultimately form the ejaculatory duct, you can find out here, and that ejaculatory duct is draining at the level of urethra. So this is the common ejaculatory duct and urethra, which is released externally at the tip of the penis. Now apart from these main parts, certain other parts are also associated with this male reproductive system, particularly this seminal vesicle as well as this prostate, which is located here, right? So this seminal vesicle and the prostate, they are going to secrete various components which are part of the semen, right? But semen means we all know that sperms is present inside the semen and sperm production occurs inside this testis. So that's why the testis is the main part of the male reproductive system. On your right hand side, a detailed diagram of the testis internal structure is given, where you can find out that there are presence of coiled tube inside this testis. We all know that this coiled tube are known as the seminiferous tubule. It is said that inside a single testis, around 900 such seminiferous tubules are present, right? Now, both end of the seminiferous tubule, what I told, that the both end of this seminiferous tubule, they are connected with this rete testis, and that rete testis is ultimately draining at the level of the epididymis, right? Now, each of this part has different function. Like for example, if I ask you what is the function of this rete testis, so at the level of this rete testis, there will be reabsorption of excess fluid for the concentration of the sperm. What is the meaning of that? Means when the sperm production occurs at the level of this seminiferous tubule, they are the diluted sperm. Now from that diluted sperm, you have to reabsorb huge amount of fluid, and then only the sperm will be concentrated. This reabsorption of fluid occurs at the level of rete testis under the influence of one hormone, that is estrogen. So this can be asked in a future MCQ, so remember this. Then that concentrated sperm will enter at the level of the epididymis. When this sperm, concentrated sperm along with the secretory fluid is passing through this epididymis, there will be certain changes inside the sperm that we are going to discuss, right? So first of all, we are going to discuss the functions of this seminal vesicle and the functions of this prostate, and then we are going to discuss what is the functions of this epididymis, and then we will reach at the level of this seminiferous tubule to understand that how the spermatogenesis or the formation of mature sperm from spermatogonia, that is spermatogenesis, how that is happening at the level of this seminiferous tubules, right? So first, let's discuss what is the function of these two, right? So seminal vesicle and prostate. So seminal vesicle, it secretes a mucoid secretion with high amount of fructose, citric acid, prostaglandin and fibrinogen. Now among different components, this prostaglandin is one of the important components which makes the cervical mucosa, as it is written here, more receptive from the sperm, right? So whenever this prostaglandin is secreted, or when the semen is ejaculated at the level of the vagina and female reproductive tract, the prostaglandin component of the semen, which is secreted from the seminal vesicle, it is making the female reproductive cervical mucosa more receptive for the sperm. It will take the sperm more avidly, more vividly, right? Now also it causes reverse peristalsis movement of the uterus and the fallopian tube, so that the sperm will be easily transported from the level of the vagina up to the level of the isthmus, right? Now apart from this seminal vesicle, look at the function of the prostate. So prostate secretes various components. The main component is the zinc, right, which has already been asked in MCQ, that zinc is secreted by which of the gland? Prostate. Along with that, it also contains huge amount of calcium ion, phosphate ion, a clotting enzyme as well as profibrinolysin. Now, slight alkaline secretion of the prostate helps in sperm motility, although the optimum motility of the sperm occurs at pH of 6 to 6.5. Right. So, these two accessory glands, they are secreting the various components of the semen. It is said that seminal vesicle generally secretes 60% of the total volume of the semen, and prostate glands secrete 20% of the total volume of the semen. So, this is a function of this seminal vesicle and prostate. Now, again we will go back to this original diagram. So, we have discussed already the function of the seminal vesicle and prostate. Now, next thing we are going to understand that this testis, which is connected with this epididymis, both of this structure is located outside the abdominal cavity inside the scrotum. We all know that. So, why these testes are located outside the abdominal cavity? You know that inside this abdominal cavity, the temperature is very high. In high temperature, the sperm production at the level of seminiferous tubule will be defective. It has been already proven that if the testis is placed inside the abdominal cavity, it will not be able to produce adequate amount of sperm. So, this testis is located outside this abdominal cavity, as well as there is a mechanism inside this testis by which the temperature of the testis remain in a low condition in comparison to the abdominal temperature, and that system is known as the counter current exchange system at the level of testis. Right. So, look at. So, this is your testis. Okay. It is supplied by this testicular artery. So, this testicular artery which is coming from this abdominal cavity. So, because it is bringing blood from the abdominal cavity, the temperature of this blood is very high, and that is at the core body temperature near equal to 37 degrees centigrade. Now, if this blood is directly entering at the level of this testis, then it will causes a problem in sperm production. So, whenever this blood is going towards the testis, it will exchange its high temperature. Okay, so it will exchange some heat to the level of these venous plexus which are present surrounding this artery, and this venous plexus is known as the pampiniform plexus. So, whenever this exchange of heat is going from this artery towards the venous plexus, the temperature of this testis will be lower than that of the abdominal cavity, near equal to 32 to 35 degree centigrade. So, look at here, this diagram has been simplified on your right hand side. So, this is the testicular artery which is bringing blood towards the testis, and this is the pampiniform plexus, right, the venous plexus of the testis. Whenever this testicular artery is bringing the blood towards the testis, there is exchange of heat from the artery towards the vein, and these two tubes, they are parallel to each other, right, and they are very close to each other, and the flow of blood is opposite to one another. That's why this whole system is known as the counter current system, and here is exchange of the heat between these two systems. That's why this is counter current exchange system, and because of this counter current exchange system, I can find out that the temperature inside the testis will remain 32 degrees centigrade to 35 degrees centigrade, which is lower than that of the core body temperature, which this lower temperature inside the testis will be helpful for sperm production. Right. So, this way you have to remember that why this counter current system is important for sperm production or spermatogenesis. Now, next we are going to discuss that along with this testis, we can find out that this epididymis is attached, and I told you that epididymis has some important functions. So, when the sperm is reaching at the level of epididymis and when it is passing from the head towards the tail of the epididymis, what are the changes that is happening? This is one of the important thing that is important for your MCQ, right? So, look at first of all, the sperm remain inside the epididymis for 12 to 26 days, right? So, during this staying, certain important changes occurs inside the sperm. Number one, it acquire motility. What kind of motility? There are different kinds of motility, but the motility which is helpful for fertilization is known as progressive forward motility. Progressive forward motility is acquired at the level of epididymis. Not only that, the sperm become almost fully mature at the level of epididymis. Right. So if you aspirate the sperm from the level of the epididymis, that is capable of fertilization. But normally when the sperm is ejaculated at the level of vaginal tract or female genital tract, there is a process which is known as the capacitations, right, by which sperm become hypermotile. And this capacitations or hypermotility of the sperm is essential for fertilization. That's why it is said that although the sperm is mature at the level of epididymis, full maturity occurs at the level of female genital tract, right? So this you have to remember. That final motility or the full maturity of the sperm means the process of capacitations which is going to happen inside the female genital tract, right? Okay. Then maturation of the acrosome, that at the head of the sperm there is a cap which is known as the acrosome, which contains certain enzymes which is helpful during the process of fertilizations. This synthesis and maturation of the acrosomes also occur when the sperm is staying inside the epididymis. How many days it is staying? 12 to 26 days. So there is a long stay of sperm inside the epididymis, and during this stay many important changes are occurring inside the sperm, right? Along with that, there will be decrease in cytoplasm and cell volume, and the sperm will have the ability to bind to Jena pellucida, because all of ultimately target of the sperm is to fertilize the ovum. For the fertilization, the sperm has to bind with the zona pellucida, and then it will penetrate inside the ovum. So this binding capacity, ability to bind the zona pellucida, this also occur at the level of epididymis, right? So all of this thing is happening inside the epididymis. Now the most important thing that is happening here, acquisition of the progressive forward motility. This motility, how the sperm become motile, you have to remember the name of one channel which is known as CAT, SPER. CAT stands for calcium channel of sperm. That's why the channel which is responsible for giving motility to the sperm is known as CAT spur channel. So if I ask you how the sperm is acquiring this motility at the level of epididymis, you have to remember the name of one important channel, that is a CAT spur channel. This channel is basically a calcium channel, right? Because of presence of this channel, calcium enter inside the sperm and sperm become motile. So please remember the name of this channel, which is known as CAT spur channel, right? So this is what is happening inside the epididymis. After this, the sperm is fully motile and fully mature. It will be entering inside the differences, and from there it will be ejaculated out, right? Now, after discussing the function of these accessory parts, now we are entering what is happening inside the testis, right? Means what is happening inside the seminiferous tubule. So if you take the cross section of the testis, you are going to get the cross section of the seminiferous tubule. So suppose this yellow color diagram that I am drawing here, this is the cross section of the multiple seminiferous tubules. In between the seminiferous tubules, the interstitial tissue of the testis will contain certain cells. These cells are known as the Leydig cells. So this is nothing but your Leydig cell, right? So all of us know that it is the cells which is going to secrete your testosterone. So where these Leydig cells are located? Leydig cells are located in the interstitial tissue of the testis. That's why we say that interstitial cells of Leydig, right? Now, apart from this Leydig cell, obviously there will be presence of certain blood vessel that you also know, right? So the blood vessels are also present here. Now, if you look inside the seminiferous tubule, the major cell component which you are going to find out here is long type of cell, which is nothing but Sertoli cells. So there will be presence of long cells inside this seminiferous tubule, and these cells are nothing but your Sertoli cells. In between Sertoli cells, obviously there will be presence of different stages of different stages of spermatogenesis cells, right? So the spermatogenesis is occurring inside this seminiferous tubule. So the different stages of this spermatogenesis cell, like for example, spermatogonia, primary spermatocyte, all of them, they will be located close to this seminiferous tubule, right? So this is a basic diagram of the cross section of the testis. So this diagram has been given in Genong. So this is I have taken from Genong. So first of all, you note down the presence of the basal lamina, right? So this is the basal lamina which is lining the seminiferous tubule. So this is your basal lamina. So inside this basal lamina, you can find out that this is the lumen of the testis. So inside this lumen, what is the major cell that you are finding out? This is here your Sertoli cell, right? So this pink colored cell which is present here, this is nothing but your Sertoli cell here. And close to this Sertoli cell, various stages of spermatogenesis process, like for example, this is spermatogonia, right? Then the primary spermatocyte cell, then the secondary spermatocyte cell, and the fully mature sperm. They are all attached with this Sertoli cell only. That's why we say that Sertoli cell is like that of the mother in the process of the spermatogenesis. All the children, they are very close to the Sertoli cell, and the Sertoli cells not only gives nutrition to this developing sperm, other supports are also given by this Sertoli cell. So it's like mother, right? So the Sertoli cell, close to the Sertoli cell, there will be various stages of the cells which are responsible for spermatogenesis process. Now, apart from this, you have to remember that cytoplasm of the two Sertoli cell. So suppose this is one Sertoli cell, okay? This is another Sertoli cell. Cytoplasm of the two Sertoli cell, they are also connected with one another. Like for example, suppose this is cytoplasm of one Sertoli cell, this is cytoplasm of one Sertoli cell. They will extend their hand, right? And they will be connected with one another. If you note down this diagram here also, you are going to find out this connection between two Sertoli cells. Look at here, okay? Look at here, the diagram from Genong. So look at this pink colored cytoplasm of the Sertoli cell, right? So here, this is pink colored cytoplasm. This is the pink colored cytoplasm from another Sertoli cell. Both of them are connected with one another. So this is the connection between two Sertoli cell which I am talking about, okay? Again, you can find out here, this is the connection between two Sertoli cell which I am talking about. So, whenever these two Sertoli cells are connected with one another, it's forming a barrier, and this barrier is known as the blood-testis barrier. So, how this barrier is formed, we are going to understand this a little more detail. So, look at my diagram, the simplified diagram. So, this is one Sertoli cell, right? Okay. This is your basal lamina, right? And this is another Sertoli cell. Now these two Sertoli cell, the cytoplasm of them, so cytoplasm of one Sertoli cell is extending its process, cytoplasm of another Sertoli cell is also extending its process, and these two cell cytoplasm are connected with one another. So there is a attachment between two plasma membrane with one another with the help of a molecule which is known as tight junction. So what is happening? One Sertoli cell is extending its process, another one is also extending its process. These two processes are attached with one another with the help of a protein molecule that is known as tight junction, and whenever this tight junction is attaching both the Sertoli cell, it is forming a barrier. So there is a barrier here, and this barrier is known as blood-testis barrier. So if you note down the diagram from Genong here, so this pink color one is the blood-testis barrier. Which cell is located outside this blood-testis barrier? So all these cells, they are located inside the blood-testis barrier, and the cell which is located outside this blood-testis barrier, this is nothing but spermatogonia. Okay, so spermatogonia is located outside this blood-testis barrier, but other stages of spermatogenic cell, like for example primary, secondary, early spermatid and mature sperm, all of them are located inside the barrier. So look at here. So this is your Leydig cell, interstitial cells of Leydig. This is your capillary. This is your blood capillary. So I told you already that spermatogonia, it is located outside this blood testis barrier, but all other cell stage, like for example primary spermatocyte, secondary spermatocyte, okay, spermatid and mature sperm, all of them are located inside the blood testis barrier. Now the component or the compartment which is inside this blood testis barrier, this compartment which is inside this blood testis barrier, this compartment which is inside this blood testis barrier, this is known as adluminal compartment. This is known as adluminal compartment, right? And the compartment which is located outside this blood testis barrier, this is known as the basal compartment, right? So basically we can understand from here that the blood testis barrier is dividing the lumen of the seminiferous tubule into two compartments. One is the inside compartment, which is adluminal component, and one is the outside component, which is the basal compartment. Now the composition of this adluminal compartment is very different from basal compartment. So this adluminal compartment contains huge amount of potassium. Not only that, it contains huge amount of testosterone hormone which is required for sperm production, as well as is also contain huge amount of estrogen, right? So all these things are very high inside the adluminal compartment. Now look at here, I told you testosterone is very high inside the adluminal compartment, but where from the testosterone is produced? The testosterone is produced from the Leydig cell which are present outside this adluminal compartment. But because the testosterone is an steroidal hormone, it can easily penetrate this blood testis barrier and it can enter inside this adluminal compartment easily. So that's why the concentration of this testosterone is very high inside this adluminal compartment. But the problem is that this testosterone can again return back to the blood, but this returning back will be prevented by secretion of a binding protein for the testosterone. So what I am saying, this Sertoli cell will produce a binding protein that will attach with the testosterone and it will prevent the backflow of testosterone from this adluminal compartment to the blood or the basal compartment again. Getting my point? So this way the testosterone concentration remain high inside this adluminal compartment. Now second, I told you the estrogen concentration is also very high. Where from this estrogen is coming? This Sertoli cell contain high amount of aromatase enzyme activity, and this aromatase enzyme, they can easily convert the androgen into estrogen. That's why the huge amount of estrogen will also be present inside this adluminal compartment. Now, what will be the function of this testosterone and estrogen in the spermatogenesis? That we will look at the end of this chapter. But before that, so what we have to remember that the blood testis barrier, right? Blood testis barrier. What is the function of this barrier? It is protecting this spermatogenic compartment. So the spermatogenic compartment is the adluminal compartment. So blood testis barrier is protecting this compartment from the toxins of the blood. We know that various toxins are present at the level of blood which can hamper the spermatogenesis process, and from these toxins this blood testis barrier is protecting the adluminal compartment. Not only that, the other way is also true. What is the meaning of that? When the spermatogenesis is occurring inside this adluminal compartment, there will be production of certain antigens, and those antigens, if they are entering from this adluminal compartment into the blood, then serious immunogenic reaction can happen, and that immunogenic reaction is again prevented by blood testis barrier. So basically blood testis barrier is acting in two ways. It is preventing any toxin from this blood, preventing this any toxin which is entering from blood to the adluminal compartment, is preventing that, and the other way is also true. It is preventing any antigenic component from adluminal compartment to blood. So this way blood testis barrier is very important. So if I ask you the last question, that who forms the blood testis barrier, your answer is Sertoli cell, obviously, along with uricare tight junction. That is, this is forming the blood testis barrier. So what is the function of this Sertoli cell? The Sertoli cell has many functions. Certain functions we have discussed and certain functions are yet to be discussed. So look at here. First, it forms the blood testis barrier. We have discussed that, and I told you it's like a mother for the process of spermatogenesis. So the nourishment of the sperm and the protect the developing sperm. Okay, this is also done by Sertoli cell. Now, the important MCQ that has been asked, that which receptor is present on the Sertoli cell? FSH receptor as well as the androgen receptor is present on the Sertoli cell. Neither this FSH or the androgen receptor is present on sperm. So during the process of spermatogenesis, if I ask you what is the role of FSH, so FSH is not directly acting on the spermatogenic cell. Okay, it is acting at the level of Sertoli cell, and that Sertoli cell is helping in the process of spermatogenesis, right? Then second thing, it produces androgen binding protein. I told you already that how the testosterone, after entering inside the abdominal compartment, why this testosterone is not returning back to the blood once again. It is prevented by binding of this testosterone with a binding protein, and who is secreting that binding protein? Sertoli cell. Then Sertoli cell also expresses aromatase enzyme. I told you that, and this aromatase enzyme is going to convert testosterone into estrogen. Okay, estrogen has separate role, testosterone has separate role for the process of spermatogenesis. I'll tell you. Okay, not only that, it produces Mullerian inhibitory substances in the fetal life, and that causes regression of the Mullerian duct. And the last one is also secreting, Sertoli cell is also secreting inhibin B, and this inhibin B will give a negative feedback effect on FSH, right? So there is a negative feedback process, right? How this negative feedback process is occurring? Inhibin on the important component which is giving negative feedback to FSH secretion, right? So now look at what is the function of Leydig cell. So Sertoli cell, major function is Sertoli cell, and what is the function of Leydig cell? Only one thing I have to remember, that Leydig cell is responsible for secretion of testosterone. But if you look at the function of the Leydig cell, it is similar to the adrenal cortex, particularly the zona reticularis of the adrenal cortex. We have also seen during the endocrinology that zona reticularis of the adrenal cortex, they produces sex steroid, particularly the DHEA, that is dihydroepiandrosterone, and androstenedione. But adrenal cortex cannot convert them into testosterone because it lacks the enzyme which is converting from this DHEA to androstenedione and from androstenedione to testosterone. It lacks those enzymes. So these Leydig cells, they are similar to zona reticularis, but this Leydig cell can efficiently convert the dihydroepiandrosterone to testosterone, right? Which is not possible at the level of zona reticularis. And what is the raw material for the synthesis of this testosterone? Obviously cholesterol. Okay, this Leydig cell, either they can synthesize cholesterol within them, that is the de novo synthesis, or they can take the cholesterol from the blood, right, through LDL and HDL receptor, that is known as a scavenger receptor. So for the raw material for generation of this testosterone, either it can be synthesized within the Leydig cell itself, or it can take the cholesterol from the blood. And by taking all these components, the testosterone will be produced by Leydig cell, and that will diffuse inside the seminiferous tubule into the adluminal compartment, and the aromatase enzyme of Sertoli cell will convert this testosterone into estradiol, most small amount of estradiol, right? Sperm expresses estrogen receptor. So previously I told you sperm does not contain androgen receptor, does not contain FSH receptor, but sperm contain estrogen receptor. And I'll tell you what is the function of this effect of estrogen on sperm, right? It helps in acrosomal biosynthesis. Estrogen helps in acrosomal biosynthesis at the level of sperm, right? So after discussing the various cells, that is the function of Sertoli cell, functions of Leydig cells, okay, now we are going to understand that how the hypothalamus is controlling the testis. Okay, secretion of hormone from testis, how it is controlled by hypothalamus. This is very, very simple. So the hypothalamus is going to produce GnRH, gonadotropin releasing hormone, which is produced from hypothalamic nucleus. This GnRH is going to act at the level of pituitary. Obviously, all of you know that at the level of pituitary. Okay, so when it is acting at the level of pituitary, so this is coming from hypothalamus, right? Okay, pituitary will produce two hormones, that is LH and FSH, right? Now this LH, we have seen, okay, that LH is going to act on Leydig's cell, okay, and FSH is going to act on Sertoli cell. Now this Leydig cell is going to produce testosterone, and the Sertoli cell is going to produce one of the important component, that is inhibin. Now this inhibin has negative feedback on this FSH, and testosterone has negative feedback on LH, right? This is the very simplified diagram. Now apart from this, we also know that Sertoli cell is responsible for nourishment during the process of spermatogenesis. So it is helping in the process of spermatogenesis, right? This testosterone will also help in the process of spermatogenesis. Apart from that, the other functions of Sertoli cell we have already seen, that it is going to produce androgen binding protein. It has aromatase activity by which it will also convert, or it will also produce little amount of estrogen inside this adluminal compartment of seminiferous tubule. So this way, the hypothalamic pituitary axis is controlling the testes for the production of two important hormones. One is testosterone and other one is the estrogen inside the inside the seminiferous tubule, right? Now after discussing of all of this, now we are going to see how the spermatogenesis is occurring, process of spermatogenesis, okay, how the sperm is produced from spermatogonia cell. Now I believe all of you know this process from your plus 2 level knowledge or from your PMT knowledge also, that spermatogenesis is occurring inside the seminiferous tubule. And what are the different stages? The spermatogonia cell, right? The spermatogonia cell, spermatocyte, that is primary and secondary, then spermatid, then spermatozoa. All of this cell, they are very close to this. This is very, very important. They are very close to this Sertoli cell. I told you already repeatedly that Sertoli cell is like mother. So whenever such cells are developing from one stage to other stage, all these cells, they remain attached with the Sertoli cell. Okay. Even when the mature sperm is produced, that sperm will also remain attached with the Sertoli cell. Then when you require, the sperms will be separated out from Sertoli cell and it will be entering into the epididymis. That is the whole process. Now, how step by step fashion this process is occurring inside the seminiferous tubule? What are the different stages? You know that if you open genome, it is written that one spermatogonia cell can give rise to 512 sperm. So how that is happening, right? I'll tell you. Okay. So first of all, look at different stages of spermatogonia. This is known as spermatogonia A1. This is spermatogonia A2. This way, different stages are there. Means initially, two spermatogonia, they will start division. So this is two spermatogonia. They will start division. And which stage of spermatogonia there? There's spermatogonia A1. Then A1 will be converted into A2, that will be converted into A3, that will be converted into A4. Then it will form spermatogonia, IN spermatogonia. Then it will form spermatogonia B, then primary spermatocyte, secondary spermatocyte, and then mature sperm will form. So different stages has been written here. So if you calculate, so suppose this is one cell, this is one spermatogonia cell. I told you that initially the two cell will start division simultaneously. So whenever this one cell is dividing into 2, then it will divide into 4, then 8, then 16, then 32, then 64, then 128, and then 256. So one spermatogonia cell A1 is giving rise to 256 sperm. So the other one will also divide and it will also give rise to 256 sperm. So in combination, the total 512 sperm is produced from a single spermatogonia, right? But do you think this information is true for human? Answer is no, no, no. So the statement which is given in genome, that statement is not true for human, that one spermatogonia can give rise to 512 sperm. This is true for rodent. Okay, in case of rodent, right? One spermatogonia can give rise to 512 sperm, but this is not true for human and in primates, other primates also. So how many sperms are formed from a single spermatogonia in case of human? That is the main question. Look at what I have found out from Nobel and Nell textbook of reproductive physiology. They have compared the three types of different spermatogonia. One is happening in the rodent model, one is in other primate, and one is in human. So as you can find out here, the all different stages, right? Spermatogonia A1, A2, A3, A4, intermediate, then spermatogonia B, then all these stages I have already shown you. And by this process, 512 sperm will form from a single spermatogonia. That is true for rodent. In case of primates as well as in case of human being, all of these steps which are occurring here, these steps are not there. You can find out here there is a big gap. This A1, A2, A3, all these stages of mitosis, they are not going to happening in case of human. That's why it is said that in case of human, this spermatogonia cell, they remain patchy inside the seminiferous tubule. They are not continuous. Okay. If the cells are dividing like this, one after another, they will remain connected with one another and it will be a continuous process. But in case of human and in case of primates, different stages which are occurring here, A1, A2, A3, all of these stages are absent. Because of which, in case of human, the spermatogenic process is very, very simple. It is forming the spermatogonia B, then primary spermatocyte, then secondary spermatocyte, and then spermatid or sperm. By this process, one spermatogonia is giving rise to only 8 to 32 sperm cells. On an average, your answer will be 16. Getting my point? So if the question is coming that one spermatogonia cell in case of human gives rise to how many sperm, your answer on an average 16, but exact it is 8 to 13 is the range, right? So please remember that in case of human, the steps are very, very simplified. The same thing has been given in Guyton. Right, so you can find out here the primordial germ cell, right? Primordial germ cell, that is the undifferentiated germ cells which are identified at the level of yolk sac in and around 2 to 3 weeks of gestations. They will migrate to the level of testis, right? So it is migrating, okay, and then it is forming the spermatogonia. Right, so they will migrate to the sex cord and those sex cord will be converted into seminiferous tubule. So whenever they are migrating to the level of testis, then it is known as spermatogonia. Then this spermatogonia, okay, now it is ready for division. First it will form the primary spermatocyte, then secondary spermatocyte, then spermatid, and then it is forming the mature sperm. By this process, one spermatogonia cell is giving rise to on an average 16 sperm cells. Now this whole process required total of 74 days, right? Another things you also have to remember that I told you that the spermatogonia cell, they remain outside this blood testis barrier. So whenever this spermatogonia cell is giving rise to primary spermatocyte, it has to cross through this blood testis barrier. So this spermatogonia cell, they are outside the blood testis barrier. Whenever they are dividing to primary spermatocyte, this barrier will open up for a certain period of time and the cell will move through this barrier. right? So this is the whole process of spermatogenesis. Now, two terms are written here. One is spermiogenesis and one is spermiation, right? So spermiogenesis, the first one, spermiogenesis means when the spermatid is converted into mature sperm, means this last stage which is requiring this 21 days according to Guyton, this is known as the spermiogenesis. So spermiogenesis means spermatid is converting into mature sperm. The whole process is known as spermatogenesis, but spermiogenesis means the last steps where the spermatid is converting into mature sperm. Now I told you that even after formation of the mature sperm, the cells will remain attached with the mother cell. And who is the mother cell? Sertoli cell. So you have to release the mature sperm from the Sertoli cell, and then only the sperm will able to start its journey towards the vagina, right? So the release of sperm from Sertoli cell is known as spermiation. So this spermiation means it is nothing but release of sperm from Sertoli cell, right? Sertoli cell. After release, now it will enter at the level of epididymis and its journey started, right? So this is the whole thing that you have to remember. Now during this whole process of spermatogenesis, what are the hormones that you require, right? Bottom line of this is that you have to remember two major hormones. One is FSH and one is testosterone. Third hormone is the estrogen. But first of all, you remember FSH, and second one is the testosterone. If I ask you which FSH acts in which stage of this spermatogenesis, your answer will be FSH is required for the early development of this spermatogonia cell. So during this process of spermatogonia dividing into primary spermatocyte, secondary spermatocyte, during this process you require more amount of FSH. But testosterone is particularly required for the terminal stages of spermatogenesis, means this spermiation process, this release process, all of these things require the presence of testosterone, right? So early stages spermatogenesis cell, FSH is the required. Late stage spermatogenesis, the testosterone is the main hormone. So look at the functions of testosterone, right? I told you already the process of spermiogenesis, right? Spermiogenesis means from spermatid to mature sperm, that is spermiogenesis. Spermiation, the release of sperm, that also requires testosterone. And the meiosis, right? The process of meiosis is also require the help of testosterone. But if you look at the FSH is mainly for proliferation of the Sertoli cell, which is the mother for the spermatogenesis process. Not only that, the spermatogonial cell proliferate, okay, and it proliferate up to the level of spermatocyte stage. This proliferation is under the control of FSH. So when the spermatogonia cell is proliferating, and this proliferation is going on up to the level of primary spermatocyte, then secondary spermatocyte is formed. But up to the level of this primary spermatocyte's proliferation, FSH is one of the major hormone, right? And it also supports the meiosis process. So meiosis is requiring the double support. The testosterone is also required, FSH is also required. But testosterone is main hormone for terminal stage of spermatogenesis, and FSH is for early stage of spermatogenesis. Now what about estrogen? I told you already that testosterone converted into estrogen under the influence of aromatase enzyme, which is present inside the Sertoli cell. Estrogen does two important functions. One is acrosome biosynthesis, and second one, it reabsorbs the fluid, okay, and concentrate the sperm. I told you that when the sperm is produced from at the level of testes, it enter at the level of rete testes, right? So from seminiferous tubule, it is entering at the level of rete testes, and at the level of rete testes, huge amount of reabsorption of fluid is there. Because of the reabsorption of fluids, concentration of the sperms occur. And which hormone helpful for this reabsorption of excess fluid? That is estrogen. And estrogen's second function is acrosome biosynthesis. That's why the sperm contain the estrogen receptor, because this estrogen is helpful for acrosomal biosynthesis, right? So male hormone which is requiring for this spermatogenesis process is testosterone, okay, which is synthesized from Leydig cell. Now the testosterone, it has some important function which is done by itself, testosterone itself. And testosterone can be converted into another variety, that is dihydrotestosterone, by an enzyme that is 5-alpha reductase. This dihydrotestosterone has certain other important functions. And testosterone can also be converted into estradiol or estrogen by the enzyme aromatase. This estradiol also has certain function on male. So what is the function of testosterone in male? Right, so it acts on androgen receptor obviously, and it is responsible for mainly development of internal genitalia. So remember, testosterone is for internal genitalia and dihydrotestosterone is for external genitalia development, right? That's why it is written testosterone has main action on internal genitalia. Not only that, for growth of bone, for growth of skeletal muscle, and for erythropoiesis, all of these require testosterone. Dihydrotestosterone also act on the similar kind of androgen receptor, and the action of this dihydrotestosterone is mainly on external genitalia and hair follicle. And what about estrogen in male? Estrogen is responsible for closure of the epiphysis, so it is acting at the level of bone, and it also has effect on libido. What is that effect? Look at, so this is the detailed action of testosterone in male. So first one, okay, look at here. Testosterone is responsible for maturation of the Wolffian duct structure and the formation of the male internal genitalia during development. What is that internal genitalia? The epididymis, the seminal vesicle, the vas deferens, all of them. But if you compare it with the dihydrotestosterone, it is development for the male external genitalia, particularly the penis, the scrotum, and the prostate. Right? Very important. I'm telling you. Now, not only that, if you look at the second function here, the pubertal growth is also done by testosterone. Depending on the voice, increasing muscle mass and bone mass, that is also done by testosterone, right? Growth of the penis and seminal vesicle growth, right? Not the differentiation, not the development, and increase in RBC number as well as the process of spermatogenesis. Rather than testosterone, if you look at the dihydrotestosterone, you find out here that what is the function? It is responsible for growth of the prostate and penis at the time of puberty. It is responsible for development of facial hair, acne. It increases the sebaceous gland activity, as well as male pattern of baldness is also under the control of this dihydrotestosterone, right? Now the last one, the function of estradiol, it is responsible for fusion and closure of the epiphysis. Please note down, right? If you prevent the conversion of estradiol from testosterone, right, what will happen? There will be increased height of the person, because the epiphyseal closure is not going to happen, which is done by estrogen. So please note down, the testosterone itself is responsible for the growth of the bones and muscle, but the closure of the epiphysis is done by conversion of the testosterone to estradiol, right? As well as estrogen also increases, okay, administration of estradiol to a man increases the libido. That's why I told you that estrogen also contains the libido of male person. So this is all about the male reproductive physiology, which you have to read for your MCQ purpose.